Right now, inside every single one of your cells, something pretty wild is happening. They're eating parts of themselves. And it might sound like science fiction, but this self-eating, which is called autophagy, is actually one of the most important cleanup and recycling systems that keeps us running. So today, we're going to dive right into the cell and see how this incredible microscopic housekeeping actually works. So, here's our game plan. We'll kick things off by explaining what this self-eating thing really is. Then we'll follow the entire construction of the cell's recycling bin, from the very first spark all the way to the final delivery. We'll also take a look at the master switches that turn the whole system on and off. And then, at the end, we'll connect it all back to why this is so critical for our health.
All right, first up, let's get a really clear idea of what we're talking about when we say self-eating. Okay, so the official name for this is macroautophagy. Just think of it as the cell's ultimate recycling program. It's not just about tossing out old or broken parts, it's about carefully dismantling them into reusable building blocks, like amino acids. This is absolutely essential for keeping everything inside the cell stable and balanced, a state that scientists call homeostasis.
And this is the key takeaway here. This is not some kind of cellular self-destruction. It's actually a super meticulous housekeeping process. The cell is just tidying up, breaking down old furniture to get the materials to build new stuff, making sure everything runs as smoothly as possible. So how does this whole thing even begin? Well, like any big construction project, you need a signal to get started and a management team to get the ball rolling. And inside the cell, this very first stage is called initiation. The entire process can really be broken down into these 6 key stages. And it all starts right here with initiation. That's the moment the cell basically says, all right, it's time. Let's build a recycling bin. So who makes that call? Well, that decision comes down to a management team of proteins called the ULK1 complex. The boss is a protein called ULK1. Think of it as the lead foreman on the job site who gets the go signal. ULK1 then activates its crew. You've got ATG13, which helps ULK1 get the whole process started. Then there's FIP200, which is this big scaffold protein. It's like the platform that holds the whole team together in the right place. And finally, you have ATG101, another essential partner that keeps the whole complex stable so that assembly can begin. Together, they create what's called a pre-autophagosome, basically the first blueprint for our recycling container. With the project officially greenlit, it's time to mark the construction site and actually start building the walls of this smart garbage bag that's going to collect all the cellular junk. We're now zeroing in on nucleation and elongation. These are the 2 stages where the physical structure, the autophagosome, actually gets built. This is where the real construction happens. The first crew to show up are the surveyors, and they get their orders from the foreman, ULK1. So ULK1 flips the switch on a protein called Beclin-1. Beclin-1 then grabs its partners. The most important player here is VPS34, which is a special enzyme that creates a lipid molecule called PI3P. Now, this PI3P is basically the surveyor's tape. It's the X marks the spot for exactly where the construction needs to happen. The third partner, VPS15, is there to stabilize the whole complex, making sure the surveyors can do their job right.
So once that PI3P marker is laid down, it acts like a landing beacon. Proteins called whippies are drawn right to it, and they're the signal that calls in the rest of the construction machinery. At the same time, you've got this specialized cleanup crew, cargo receptors like P62 and MBR1. Their job is to find and grab onto any cellular waste that's been tagged for disposal, usually with a little tag called ubiquitin. And they start piling it all up at the construction site ready for pickup.
All right, now for the builders, and they actually work in 2 teams that are linked together. First, you have the ATG5, ATG12, ATG16L1 complex. This huge structure shows up and figures out exactly where the membrane wall should expand, and it helps bring in the second team. Now, the second team is the LC3 system, and it handles the most important building block. It all starts with a protease called ATG4, which snips a protein to make its active form, LC3I. Then, in a little 2-step process, an enzyme called ATG7 activates this LC3I, and another enzyme, ATG3, finishes the job by sticking it directly onto the growing membrane. This is literally what builds the wall, brick by brick. And this right here, this is probably the most critical transformation in the whole process. When that LC3I protein gets attached to the membrane, its name changes to LC3II, and that change is what physically makes the membrane grow and curve and expand, slowly wrapping around all the cargo. In fact, the presence of LC3II is the absolute gold standard marker that scientists look for to know for sure that autophagy is happening in a cell.
Okay, so the cargo is now completely sealed up in this double membrane bubble called the autophagosome. The package is ready. Now it's time for delivery to the cell's ultimate recycling center, the lysosome.
We are officially on the home stretch, the final 3 stages. The vesicle matures, it travels to and docks with the lysosome, they fuse together, and then finally, all the contents get broken down into their basic parts.
So how does it find its destination? Well, the autophagosome uses a kind of molecular GPS called Rab7. It dives the vesicle through the cell's crowded interior to find a lysosome. And once it gets there, proteins called snares on both surfaces act like these incredibly powerful molecular zippers. They literally grab onto each other and pull the 2 membranes together with so much force that they fuse into one single compartment.
Once they're fused, this new compartment gets a new name, an autolysosome. And immediately, a pump on its surface called the VATPase starts flooding the inside with protons, which makes it super acidic. This acid bath then activates a whole bunch of powerful digestive enzymes, including things like cathepsins. These enzymes are the demolition crew. They act like molecular scissors and just dismantle everything inside, proteins, organelles, fats, all the way back down to their basic reusable building blocks for the cell. But, this is an incredibly powerful process. The cell can't just have this running at full blast all the time. So how does it decide when to turn this whole cleanup program on or off? Well, that's where the master regulators come in. The central control system is basically a constant tug of war between 2 master sensors. When the cell is running low on energy, a protein called AMPK gets activated. It's the go signal, and it tells the ULK1 complex to start autophagy. But when the cell has plenty of food and is in growth mode, a different protein, MTRC1, is active, and it's the stop signal. It works by shutting that exact same ULK1 complex down. It's a really simple, elegant balance between feast and famine. But beyond those immediate signals, there are other layers of control. For example, a protein called BCL2 can act like a direct brake pedal. It can just grab onto Beclin-1 and physically stop the surveyor's team from ever starting their work. And then you've got the long-term factory management. If autophagy is needed for a while, a master switch called TFEB goes into the cell's nucleus and starts turning on the genes to produce more autophagy machinery. And to balance that out, ZK-SCAN3 does the exact opposite. It's a repressor that sits on those same genes to keep the factory shut down when it's not needed.
So we've walked through the incredibly detailed molecular world of autophagy, but let's pull back from the microscope for a minute and ask the big question: why is this so unbelievably important for our overall health?
When it comes down to it, autophagy isn't just one simple process. It's right at the center of a cell's health strategy. It maintains that critical internal balance. It provides an emergency source of fuel when we're starving. It clears out toxic gunk and damaged parts before they can cause trouble. And it even defends us by capturing and destroying invading bacteria and viruses. You see, this whole process is a constant, delicate balancing act. When it's working just right, it promotes health and survival. But when the system gets out of whack, too little can lead to the buildup of toxic proteins you see in diseases like Alzheimer's, while too much can actually cause cells to destroy themselves. And this is exactly why problems with autophagy are linked to a massive range of human diseases, everything from cancer to neurodegeneration. And that leaves us with one last, really important thought. This incredibly sophisticated recycling system is running tirelessly inside us every second of every day. So the real question becomes, what happens when this fundamental process starts to fail? It's a question that researchers are intensely focused on right now, because the answers could hold the key to understanding and maybe one day even treating aging, cancer, and so much more. Let's just pull this all together. Autophagy is not some random mess. It's a super precise, highly organized quality control system. We've met the whole crew, right? From the supervisor, ULK1, who gets the first call, to Beclin-1, who preps the site, P62, who tags the trash, and of course, LC3, who builds the actual bin. And we've seen how this entire operation is beautifully balanced by the stop signal from mTOR when times are good, and the go signal from AMPK when resources are tight. This constant balancing act is just absolutely essential for keeping every single one of our cells running like it should.